Good evening, everyone. Welcome to week 10 of plant physiology. So we're on the home stretch. This is lecture 10, carbohydrate partitioning, but this is part 2. So we're going to look at physiological and ecological considerations. That is, we're going to consider some of the major photosynthetic responses of a plant to its environment, the major environmental factors and how they impact on photosynthesis. So let me move on here. All right, so you can imagine that the impact of the environment is of particular interest not only to plant physiologists but also to ecologists, agronomists, viticulturalists and horticulturalists. And we need to understand how photosynthesis responds to environmental factors such as light, carbon dioxide concentrations, temperature, as well as considering indirect responses that are mediated through stomatal closure. So, for example, effects such as humidity and even soil moisture. And of course, this is important to all of us because plant productivity and hence crop yield depends on prevailing photosynthetic rates in a constantly changing environment. So, a central question in studying the impacts of the environment on photosynthesis is how many environmental factors can limit photosynthesis at the one time? The British plant physiologist Blackman hypothesised that under any environmental conditions, the rate of photosynthesis is limited by the slowest step in the process. And this is referred to as the limiting factor. So this implies that photosynthesis can be limited by either, for example, light or carbon dioxide concentration, but not by both. So using this approach, plant physiologists will vary one factor and keep other factors constant. Now, in a leaf, 3 major processes are required for optimal performance of photosynthesis. They are rubisco activity, regeneration of ribulose bisphosphate, that is RUBP, and metabolism of those important triose phosphates. So these activities obviously take place in the mesophyll cells in the leaf, the palisade mesophyll cells and the spongy mesophyll cells as pictured here. And this describes the demand by photosynthesis in the cells for carbon dioxide as a substrate. However, the actual rate of carbon dioxide supply to these mesophyll cells is controlled by stomatal guard cells, which are located in the epidermis of the leaf. So we have these separate supply and demand functions associated with photosynthesis, and they're taking place in different cells. So it's the coordinated actions of demands of photosynthetic cells, the mesophyll cells, and then the supply by the guard cells that determine photosynthetic rate as measured by net carbon dioxide uptake. Here we have a couple of amazing images. These are scanning electron micrographs of leaves of the legume Thermopsis grown in different light environments. And we can see that the leaf grown in the sun on the left is much thicker than the leaf grown in the shade. And you can even see the palisade cells, palisade mesophyll cells in the sun leaf are much longer. So that brings us to the leaf. The primary function of the leaf being photosynthesis. So let's consider how leaf anatomy and leaf orientation controls the absorption of light required for photosynthesis. We'll see that that photosynthesis, or rather the rate of photosynthesis, is limited by various factors. And this, of course, reflects the capacity of plants to grow under different light environments. But of course, there are limits to the extent in which photosynthesis in a species can acclimate to different light environments. And under different conditions, the rate of photosynthesis is limited by different factors. So, for example, photosynthesis may be limited by low light, low carbon dioxide concentration. Too much light can be a factor, high temperatures, low temperatures, and then, of course, varying degrees of water stress. So differences in the amount and type of light means that we need a uniform way of measuring and expressing the light that impacts on photosynthesis. So let's look at these different ways of measuring light. So light that reaches a plant is a flux, and that flux can be measured in either energy or photon units. Let's consider both of these, starting with energy units. Photon irradiance can be expressed as the amount of energy that falls on a flat sensor, so this could be a leaf, of known area per unit time, expressed as watts per square metre. So that's energy units. Now let's consider photon units. Photon irradiance can be expressed as the amount of energy that falls on a flat sensor of known area per unit time, expressed as, photon irradiance is the number of incident quanta striking the leaf in moles per square metre per second, where of course you know that moles refers to the number of photons, and 1 mole of light is equal to Avogadro's constant. And there's a bit of a typo here. Did anyone pick this? 1 mole equals Avogadro's constant, which of course is 6.02 by 10 to the power of 23. So I'm sure you remember Avogadro's number from living systems. And if you don't, that's fine. Okay, so both of these units that we just looked at can be interconverted, providing that the wavelength of light, lambda, is known. The energy of a photon, E, is related to its wavelength, where C is the speed of light, that is 3 by 10 to the power of 8 metres per second. There's a bit of a typo there, so that should be 10 raised to the power of 8. H is Planck's constant, so it's just a constant, 6.63 by 10 to the power of negative 34 joules per second. And lambda, of course, is the wavelength of light in nanometers. And of course, there's a bit of a typo here too. So 1 nanometer is actually equal to 10 raised to the power of negative 9 metres. So just be aware of that, but not that you have to remember all these figures. So from this equation, they can show that a photon of light at 400 nanometres has actually twice the energy of a photon at 800 nanometres. All right, let's consider a very important term here, photosynthetically active radiation, or PAR. So this describes the wavelengths of light between 400 and 700 nanometers. And of course, these are the wavelengths of light that are important for plants, for photosynthesis. So photosynthetically active radiation, or PAR, is usually expressed as quanta. Incoming sunlight can strike a flat leaf surface at different angles, depending on the time of day, the orientation of the leaf. And when the sunlight deviates from directly overhead, the irradiance is proportional, said to be proportional to the cosine of the angle at which the light actually hits the leaf. And cosine, I wouldn't worry too much about that. This is just a function of trigonometry. So in this image, we can see on the left here, this is incident sunlight occurring when the incoming light is perpendicular to the sensor, which would be your leaf lamina, your leaf blade. And at any other angle, so we can see the light coming in here at an angle, the incident light is actually reduced by the angle between the light and the leaf lamina. So while roughly so I'll come back to that. Here we go. So how much light is there on a sunny day? Under direct sunlight, par irradiance is about 2,000 micromoles per square metre per second, and this would be at the top of a dense canopy, for example, but only about 10 at the bottom of the canopy because most of the par is absorbed by the overhead leaves. So about 1.3 kilowatts per square metre of radiant energy from the sun actually reaches the Earth, but less than 5% of that is actually converted into carbohydrates by leaves. And this is because approximately 50% of the incident light is of a wavelength either too short or too long to be absorbed by photosynthetic pigments. And of the par that is absorbed, about 15% is actually reflected or transmitted through a green leaf. All right, we know that chlorophyll absorbs light very strongly in the blue and red regions of the spectrum. The transmitted and reflected light is enriched in green, and of course, this explains the green colour of plants. But of the 85% of par that is absorbed, a significant amount is lost as heat. Smaller amount is lost as fluorescence, and this results in less than 5% of the energy being converted to carbohydrates. In this graph here, this is actually showing the spectral properties of a bean leaf. The percentages of light that are absorbed by a plant are shown here in grey as a function of wavelength down here. The reflected light is shown in green, and we've got transmitted light shown in blue. I really don't like these graphs. To me, they're a little bit difficult to read, but anyway, this is what your textbook is showing. All right, the transmitted and reflected light, so the green and the blue regions, but in particular, the transmitted and reflected light between about 500 and 600 nanometers. This is the light that actually gives plants their green colour. And you can also see that most of the light above 700 nanometers, not all of it, but much of it, is not absorbed by the plant. And we'll come back to this bit here that actually is absorbed in just a few minutes. All right, so let's now look at how leaf anatomy maximises the absorption of light. So the anatomy of a leaf is obviously highly specialised for light absorption. The outer layer of epidermis cells are transparent to visible light. And remember, most of those epidermal cells actually lack chloroplasts. These epidermal cells are often convex. And convex cells act as lenses, therefore they can focus the incoming light. So this means that the intensity of light that reaches the chloroplasts in the underlying mesophyll cells is actually many times greater than the incident light itself. Below the epidermis, we have the mesophyll cells, both palisade and spongy mesophyll. The palisade mesophyll cells are photosynthetic cells that are packed closely together, and they're shaped like pillars, as you can see down here. And they're often found forming 1 to 3 layers deep. So this is an interesting observation. So why are there multiple layers, or often multiple layers, of palisade cells? In other words, isn't all the light intercepted just by the uppermost layer of palisade cells that we can see here? In fact, they found that actually more light than expected penetrates that first layer because of 2 factors. These 2 factors are referred to as the sieve effect and light channeling. So let's look at those 2 effects. So the sieve effect is due to the fact that chlorophyll pigments are not evenly distributed right through the cell, but they're confined, of course, within the chloroplast organelles. And this results in shading between the chlorophyll molecules, and also creates gaps between the chloroplasts where the light is not absorbed, and hence the reference to a sieve. So what about light channeling? Light channeling occurs when some of the incident light is propagated through those large central vacuoles that are found in plant cells, and also through the air spaces between the cells. Spongy mesophyll cells, let's talk about these. These, of course, are below those palisade mesophyll cells, and these are large cells which are very irregular in shape, and they're surrounded by large air spaces. Now, these air spaces actually generate lots of interfaces between air and water, and these interfaces reflect light and refract light, and this randomizes the direction of travel of light, and this phenomenon is called interface light scattering. And this is important in leaves, as these multiple reflections of light increase the path length over which those photons are travelling, and this in turn increases the probability of absorption of that scattered light. And in fact, they've found that the photon path lengths in leaves are about 4 times longer than the thickness of the leaf itself. So collectively, all these properties that we've just discussed results in a greater and more uniform light absorption throughout the leaf. And of course, some environments, for example, deserts, have so much light that light is potentially harmful to leaves. So these plant leaves have special features such as leaf hairs, salt glands, epicuticular wax that increases the reflection of light from the leaf surface. So this can reduce light absorption by as much as 40%, which minimises heating and other problems such as damage to photosynthetic apparatus. All right, some plants compete for sunlight. And even leaves on the same plant. So leaves shaded by other leaves, of course, experience lower light levels and different light quality. And as a result, they have lower photosynthetic rates. Trees that have leaves high above the ground represent an outstanding adaptation for light interception, and the elaborate branching system really increases the interception of light. So, of course, very little of that PAR, that photosynthetically active radiation, penetrates the bottom of a forest canopy or any canopy, in fact. Almost all of the light is absorbed by leaves at the top of the canopy. So in this graph here, this is showing the spectral distribution of sunlight at the top of a canopy and in the shade underneath the canopy. For unfiltered sunlight, the total irradiance was estimated to be about 1900 micromole per square metre per second, whereas in the shade, in comparison, it was only about 17 micromoles. So most of that PAR was absorbed by leaves in the canopy. Okay, what about shady habitats? In these habitats, sunflecks are actually a common environmental feature. And sunflecks are simply patches of sunlight that pass through small gaps in the canopy. And these can change the photon flux that's incident on a leaf more than tenfold within a matter of seconds. This is a critical source of energy that's only available for a few minutes maybe, and even then in a very high dose. So for some leaves, particularly those lower shaded leaves, these photons contain approximately 50% of the total light energy available during the day. And these leaves actually have special mechanisms to take advantage of the energy from sunflecks when they occur. Sunflecks also play a role in the carbon metabolism of lower leaves in dense crops that are shaded by upper leaves. And I'm sure that you probably experience the same the same situation in a vineyard. But of course, always correct me if I'm wrong when it comes to grapevines. So what are the mechanisms which these leaves actually use to take advantage of sunflecks? They actually exhibit very rapid responses by both photosynthetic apparatus and the stomata when the sunflecks actually appear. And this is of great interest to plant physiologists and also ecologists because they represent very specialised mechanisms for capturing very short bursts of energy. So now let's consider how leaf angle and leaf movement controls light absorption. So how do leaves influence the light levels within a canopy, for example? The angle of the leaf relative to the sun determines the amount of sunlight that's incident upon it. So for example, a horizontal leaf will obviously receive much more sunlight than a leaf that's at a steeper angle. Generally, you find that leaves at the top of the canopy tend to have the steep leaf angles, so less than the maximum amount of incident light is on the leaf blade. And it's common to see that leaf angle within the canopy decrease, that is, become more horizontal as you increase or go down in depth into the canopy. And of course, this is also species specific. Some leaves control light absorption by solar tracking. That is, their leaves continuously adjust orientation of the lamina so that it remains perpendicular to the sun's rays. And this includes many species, for example, alfalfa, cotton, soybean, bean, and lupin. And we've got some images illustrating solar tracking in lupin. So in A, this is showing the initial leaf orientation. And in B, we can see how the leaf orientation has changed after 4 hours of exposure to light coming in on an angle, as you can see by these arrows. And the movement of the leaves is usually generated by changes in osmotic potential and turgor pressure of a little region that's found at the junction between the actual lamina and the petiole, and it's called a pulvinus. All right, some plants have amazing plasticity, that is the flexibility to acclimate. So these plants can develop in response to a range of different light regimes. And this is called, and also other changes in the environment also, not just changes in light. And this is called acclimation. This is a process that I'm particularly interested in particular with regards to heat stress and heat tolerance in crop plants. So, for example, some plants acclimate so well that they can grow in either full sun or that they can grow in the shade. So this is one type of acclimation. So acclimation, of course, is a growth process in which each new leaf has a set of features, both biochemical and morphological features, that are suited to the specific environment in which that particular leaf develops. And acclimation is a very important ability, given that shady habitats receive less than 20% of PAR and deep shade habitats less than 1%. So in some plants, leaves that develop under very sunny or very shady conditions are really unable to persist when they're transferred to the other type of environment. So in these cases, the leaf will abscise and basically meaning that it just drops off. And if the plant survives, then new leaves that develop are actually better suited to the new environment. Some plants, of course, are not able to acclimate at all, so which means that they are adapted to either a sunny or a shady habitat, and they lack that plasticity required to acclimate. So they lack that ability to adapt morphologically and biochemically, and of course this all relates to gene expression. So sun and shade leaves have contrasting biochemical features. Shade leaves tend to have more total chlorophyll per reaction centre. They also have a higher ratio of chlorophyll beta A, and they're usually thinner than sun leaves. They also produce a 3-to-1 ratio of photosystem 2 compared to photosystem 1. This is in comparison to the 2-to-1 ratio of PS2 to PS1 that we find in sun plants. And this is probably because far red light, which is light at the extreme red end of the visible spectrum, so between about 710 and 850 nanometers, which is more greatly absorbed by photosystem 1 than photosystem 2. Far red light is actually more abundant in shady habitats than sunny ones. Sun leaves also tend to have more rubisco, and they have a larger pool of these protective molecules called xanthophylls. They're also thicker, sun leaves are thicker, and they have longer palisade cells. Now we're going to consider a very important slide. Here we're looking at the response of photosynthesis to light. So measuring net carbon dioxide fixation or carbon dioxide assimilation in intact leaves, as you can see here. This enables us to construct what's called a light response curve. And that's what we're looking at here. And this is a light response curve for a C3 plant. So what we're looking at here is carbon dioxide assimilation as a function of absorbed light, which you can see on this axis down here. So let's describe what's going on here. So, of course, in the dark, there is no photosynthetic carbon assimilation, but mitochondrial respiration, of course, always continues. It never stops. So as a result of mitochondrial respiration, you know that oxygen is consumed and carbon dioxide is given off. And this is exactly what happens even in plants. And this is represented as a -5 value on this axis here, which represents a loss in consumption of oxygen and a loss of carbon dioxide from the plant, rather than fixing carbon dioxide. Then as the absorbed light increases, as we increase in the amount of absorbed light, then carbon dioxide assimilation levels start to increase. And when CO2 assimilation, carbon dioxide assimilation, exactly equals carbon dioxide released through mitochondrial respiration, then this is referred to as the light compensation point. So this point right here at 0. So let's recap. The CO2 uptake as a result of photosynthesis, when that equals CO2 evolution as a result of mitochondrial respiration, this is called the light compensation point. And this value varies depending on the species. Then the absorbed light, as the absorbed light level increases, what happens to CO2 assimilation? It increases proportionally. And this indicates that photosynthesis is limited by the rate of electron transport in the light-dependent reactions, which in turn, of course, is limited by the amount of available light. So this portion of the graph that we're looking at here is referred to as light limited, and that's indicated here. All right, further increases, let's follow this curve around. Further increases in CO2 assimilation are eventually limited by a couple of factors. One, by the carboxylation capacity of rubisco in the Calvin-Benson cycle, or limited by metabolism of triose phosphates. So this part of the curve here is referred to as CO2 limited. So that's our light response curve. Let's recap a couple of main points from here on the next slide. So in the dark, no photosynthetic carbon assimilation occurs, but mitochondrial respiration continues. So carbon dioxide is released from the plant rather than being fixed. At greater light absorption levels, carbon dioxide assimilation starts to occur through photosynthesis, and this will reach a point at which carbon dioxide uptake exactly balances carbon dioxide release, and this is known as the light compensation point. It's interesting also to note that the light compensation point of shade plants are in the range of 1 to 5 micromoles, whereas in some plants, the light compensation point ranges from 10 to 20 micromoles. And so let's think of why this might be the case. Mostly because respiration rates in shade plants are very low. Therefore, only a little bit of photosynthesis is necessary to bring the net rates of CO2 to 0, that is, to reach that light compensation point. As a result of this, shade plants can survive in light-limited environments as they can achieve positive carbon dioxide uptake rates at much lower values of par than some plants. So the linear relationship that we saw in that light response curve between carbon dioxide assimilation and absorbed light, that is the light limited part of the response curve, reveals a slope that is referred to as the maximum quantum yield of photosynthesis. However, the actual quantum yield of photosynthesis in chloroplasts or cells or leaves is actually lower than, much lower, in fact, than the theoretical quantum yield. So it should be, quantum yield should be about 0.125 for C3 plants, where one carbon dioxide molecule is fixed for every 8 photons of light that are absorbed. But under today's atmospheric conditions of about 390 parts per million of carbon dioxide and 21% oxygen, the quantum yield for both C3 and C4 plants varies between 0.04 and 0.06 mole of carbon dioxide per mole of photons. So why is this actual quantum yield much lower than the theoretical yield? And we should answer that on this slide, which we do. So in C3 plants, this reduction in maximum quantum yield is caused primarily by energy loss through photorespiration. Who remembers photorespiration from our photosynthesis lecture? Does anyone remember photorespiration?
No, not really.
Not really? Anyone? Photorespiration. So does anyone remember the problem with Rubisco? Rubisco. So remember that the enzyme that fixes carbon dioxide in the Calvin-Benson cycle. Does anyone remember the problem? No? No, that's all right. Okay, so that enzyme Rubisco seems to have a problem where it doesn't just have an affinity for carbon dioxide, it also has an affinity for oxygen. So, of course, when it binds oxygen rather than carbon dioxide, this decreases the amounts of carbohydrates that are actually synthesized. It's not a very efficient process, and that process is called photorespiration. That's all right, you can go back and have a look at that. So in C3 plants, and this process of photorespiration is especially problematic in C3 plants, and this is thought to account for this reduction from the maximum quantum yield, because photorespiration actually decreases efficiency significantly in plants. All right, in C4 plants, the reduction from the theoretical maximum quantum yield is caused by the additional energy requirements of the carbon dioxide concentrating mechanism. So you might recall that there are additional steps in C4 photosynthesis, and that photosynthesis occurs in 2 different cell types, including those bundle sheet cells. And of course, this makes photosynthesis very efficient, and it all takes place with a minimum of water usage, but extra energy is required for photosynthesis to occur in C4 plants. Okay, so let's look at this graph here, another important graph. But before we do, let's just look at this information here, which is also important. Quantum yield also varies with temperature and carbon dioxide concentration because of their effect on the ratio of those conflicting reactions of Rubisco. So remember, Rubisco has an affinity for carbon dioxide, so it has the ability to carry out this carboxylase reaction that is fixing CO2, but it also has an affinity for oxygen, and this is called the oxygenase reaction of Rubisco. So below 30 degrees Celsius, quantum yields of C3 plants are higher than those of C4 plants, and we can see that in this graph here. So this graph is illustrating quantum yield here, a photosynthesis in a C3 plant represented by this red line as a function of leaf temperature, increasing leaf temperature, and a C4 plant represented by this green line here. All right, so we can see that at lower temperatures, quantum yield of the C3 plant is higher than the C4 plant here. But of course, As temperatures increase, then photorespiration increases. That is the ability of, or the affinity that rubisco has for oxygen. This increases with increasing temperature. So when photorespiration increases, then we have a decrease in quantum yield in C3 plants. Now, C4 plants are affected differently because they have that carbon dioxide concentrating mechanism in the bundle sheath cells. So they don't have such a problem with photorespiration. In other words, quantum yield in a C4 plant is not temperature dependent, and that's seen here as the quantum yield is maintained at the same level regardless of increasing temperatures. So this is actually a very useful graph for you to remember and be able to explain why at low temperatures quantum yield is higher in C3 than C4 plants, and then how this is reversed, as we can see over here, with increasing temperature. So if you can explain that, that would probably be quite useful. All right, let's look at higher photon fluxes. At higher photon fluxes, the photosynthetic response to light starts to level off and eventually reaches saturation. Hence, higher light levels beyond saturation no longer affect photosynthetic rates, which indicates that factors other than light have become limiting. So, for example, electron transport rate might be a factor, rubisco activity, or metabolism of triose phosphates. Above the saturation point, photosynthesis is commonly referred to as carbon dioxide limited, as we described earlier. And this is thought to reflect the inability of the Calvin-Benson cycle enzymes to actually keep up with the production of ATP and NADPH that arise from the light-dependent reactions. But in reality, only rarely is photosynthesis light saturated at the level of the whole plant. So individual leaves may be light saturated, but if we average that out across the whole plant, light saturation is really a problem. So crop productivity is related, of course, to the total amount of light received during the growing season, and given enough water and nutrients, the more light that the crop receives, the higher the biomass. So given that, I've wondered in the past, and obviously you can't do this in broadacre cropping, but let's say in a small vineyard, has anyone ever tried to set up something that will focus the light onto the grapevines, just on days when light levels are low? Some sort of mechanism, some sort of system, simple system that can actually reflect light onto the grapevines on certain days, and that can be sort of hidden on days where you've got good light intensity. Yeah, go on, Luke.
They use black plastic on some vineyard floors under the vines.
Oh, yes, yes.
And that can reflect light back up into the bottom of the canopy.
Ah, I wouldn't think that the black plastic would reflect light very well.
Well, it's just plastic, I think. I don't know if it's black plastic, but I have heard of people using plastic to try and increase light into canopies.
Yeah, yeah. Yeah, I've often wondered about that, because if you had a small enough, just a small vineyard, I think it's certainly something that could be tested, something that could be set up, you know, on cloudy days, for example, and something that can be set up and dismantled very easily, because obviously you don't want, you know, super reflection of incident light on really sunny days, because that would just be too much, and you probably cause more damage than good.
That's right.
Yeah. Anyway, also, just a thought. Yeah.
Yeah, and also lowering trellises it's not light so much, but I think it's radiant heat.
Yeah.
And rocks and things like that in vineyards.
Mm.
The rocks heat up and
Yeah.
can give off extra heat during the night.
Right.
It's not a light thing; it's more of a heat thing. I think that helps to get better tannin levels and things like that in red fruit.
Oh, right. Okay. So you're increasing the heat load. Is that right? Using
Increasing the heat load with a lower
using
canopy.
rocks.
Yeah, rocks.
Right.
Just radiant heat off the ground.
Right. Okay. So does that mean that you would tend not to have a lot of vegetation in between the vines, or and yeah?
Some vineyards this is in sort of France and things like that. They have really rocky sort of vineyard
Soil.
floor.
Yeah.
Rocky floor of rocky soil.
Mm-hmm.
And they have a very low like a low trellis. So it's instead of the cordon being at waist height, it might be at knee height.
Ah, right. Okay. Really low trellis.
Very low trellis.
Very low trellis. Yeah.
We've got one of our vineyards at Stonia that we've got about 30 rows that are the trellis is very low. And we use that for a whole bunch fermentation because it actually ripens the stems a bit more.
Right. Okay.
Anyway, yeah.
And is it done in that way, lowering the trellis height, so you don't have the shading effect from one row to the next?
No, well, the actual trellis height is the same. It's the actual cordon height that's lower.
Oh,
so the fruit zone is a lot lower.
Yeah, that's right. So wouldn't that sort of minimise shading from one row to the next?
Not really, because the trellis height is still the same.
Okay.
So the row height is still the same. The posts are still the same height. So
Mm-hmm.
it wouldn't really matter that side. It's more so to actually get that ground heat.
Yeah.
I think.
Yeah. Okay.
Yeah.
Oh, there's a lot to consider when you're growing grapevines, isn't there?
That's right. There's
lots of variables. Yeah.
A lot of trials you can do. Whether anyone actually finishes off the trials and works out over 10 years what the differences are. But we usually find that low trellis compared to the high trellis, which is right next door to it, is usually about a week ahead as far as sugar levels.
Okay. Yeah.
So it's riper.
Yeah. Yeah.
So that would sort of semi-prove that it works.
Well, it proves that it's actually having an effect.
Yeah, that's right. Yeah.
And a significant effect too. Yeah, absolutely. Right. Yeah, you're right. There's so many trials that you can do with grapevines. Absolutely. And we've got the perfect place to do trials, Growling Frog. Do you guys ever do anything at Growling Frog out in Yan Yean?
I've been there once. But,
yeah. Amelia, Pat, have you ever been to Growling Frog to do any work or trials with the grapevines there? No. Okay, yeah, such a great resource. I think I'm going to have to set up no time. Yeah, I know. I only went out there once, just a couple of months ago, and I thought, what a fantastic resource, so close to Epping. But anyway, yeah, of course, you've got to have students that are able to come in and spend the time.
Also, that vineyard is pretty limited because it's sort of limited because you haven't got, it's all on one aspect.
Yes, it is.
And they haven't really done anything with different pruning methods, or it's all very, I think it's all one variety too, isn't it?
I don't know. Yeah, I don't know. I was there. There was no one else around, and I was just walking around having a good look at it.
Yeah.
Noticed a few really interesting things with individual vines that were showing incredible vigour compared to most of the vines that were there.
Right.
And that was fascinating for me, wondering why this one particular standout vine versus all the rest in that particular row, or even in the adjacent rows as well.
Did you notice the drop in vigor at the top near those gum trees?
No, they all looked pretty pretty poor actually.
Yeah.
Whether they were near the gum trees or down the bottom near that little lake there.
Yeah.
There was sort of no difference that I could see.
Up near the gum trees, there's a lot of vines missing and...
Oh, yes, I did. I saw that.
Yeah.
There was a lot missing. A lot of vines missing, yeah.
And that's just that's the gum trees, like, just leaching the absolute crap out of the soil.
Yeah, I can imagine. Yeah, it certainly needs some tender loving care, that vineyard.
Yeah, it does.
It really does.
Loving care,
It does. Anyway, I suspect that researchers' dream, absolutely. I'd love to do something up there, but yes, time, as you said, Amelia, time is always a big problem. And spreading yourself too thin across multiple research projects is also a problem. But anyway, we should move on. This is a very long lecture, isn't it? All right, let's consider excess light. Excess light energy. So when exposed to excess light, leaves obviously have to dissipate that excess light energy to avoid damage to the photosynthetic apparatus. And there are several routes for energy dissipation, but the most important mechanism is thought to involve the transfer of absorbed light energy away from electron transfer to heat. Towards heat production. And this involves what's called the Xanthophyll cycle, which is not very well understood, and I certainly don't know very much about it. But they do know that there are three main carotenoid pigments involved in the Xanthophyll cycle, called violexanthin, antherxanthin, and zeaxanthin. And of course, I believe that it's because these names are so difficult to say that no one wants to work with this cycle. Only joking. So this image shows changes in xanthophyll content as a function of irradiance over the course of one day in sunflower. And as the amount of light increases, then a greater proportion of this violexanthin is converted to zeaxanthin and antherxanthin. And you can see peak levels at midday. And these molecules are involved in dissipating excess excitation energy. An alternative means to reducing excess light energy is to move the chloroplasts so that they're no longer exposed to high light. In this image, we can see cells. These are cells of the duckweed called lemna. And we've got the same cells under three different conditions: darkness, weak light, and a strong blue light. So in darkness and low light, the chloroplasts, you can see, are positioned near the upper surface of the cells. So of course they can absorb maximum amount of light. Look at the incredible difference here under strong blue light. The chloroplasts move to the side walls of each cell where they shade each other and of course minimize that absorption of excess light. Quite amazing images even here. So what is the problem with excess light energy? With excess light energy, the reaction centre of photosystem 2 seems to be particularly vulnerable. The reaction centre can be inactivated and often damaged in a phenomenon that's called photoinhibition. There are two major types of photoinhibition. This one here, called dynamic photoinhibition, occurs under moderate excess light. There is a decrease in quantum efficiency, but the maximum photosynthetic rate remains unchanged. And this is caused by the diversion of energy to heat. All right, so this process of photoinhibition, this is often temporary and it's reversible. The second type of photoinhibition is chronic photoinhibition, and this results from exposure to high levels of excess light that actually damage the photosynthetic apparatus and decreases both quantum efficiency and the photosynthetic rate. These effects can be long-lasting and they can last for weeks or even months, which is quite interesting, given that you would expect a reversal of this once the photosynthetic apparatus have been repaired, but that obviously can take some time. All right, so here we have a summary of what we've looked at so far. So before we move on, does anyone want a quick break? 5-minute break? Yes, I have a yes, and it's a yes from me. So let's break for 5 minutes. I'll see you back here at 7:40. Okay, are we all ready? Well, who can I see there? Oh, Amelia. Hello, Amelia. I've been hiding your face. I didn't know you had your webcam on. My apologies. Oh, no, you didn't have to take it off. All right. Yes, you did. My laptop actually doesn't have a webcam, which I'm not unhappy about. All right, so I think we'll get started again. Now we're going to consider photosynthesis responses to temperature. So the heat load on a leaf would be enormous if all solar energy was absorbed and no heat was lost. But this doesn't occur because leaves absorb only about 50% of total solar energy. Still, this is an enormous load, and this heat load is dissipated in several ways. One, by the emission of long-wave radiation at about 10,000 nanometers, which we can see here. Also by what they refer to as sensible heat loss. This includes both conduction and convection of heat to cool air. And then, of course, by evaporative or latent heat loss. And this is as a result of water loss from transpiration via the stomata. Okay, that's summarised here for you: radiative heat loss, sensible heat loss, and latent heat loss. So I won't go over that again. Photosynthesis is temperature sensitive. So when photosynthetic rates are plotted as a function of temperature, as we can see in this graph, for either a C3 leaf or plant or a C4 leaf or plant, under ambient carbon dioxide concentrations, the curve has a characteristic shape, or rather I should say the curves have characteristic shapes. There are two contrasting responses. The C3 plant, which we can see here, Atriplex, was grown under its natural temperate cool coastal conditions, while the C4 plant, this red line here, was grown in its natural desert conditions. So let's look at the ascending part of the curve, there and there. So this part of the curve represents a temperature-dependent stimulation of enzyme activities. So obviously the enzyme activity increases as the temperature becomes more optimal for its activity, because every enzyme has an optimum, a temperature optimum under which it works. The flat top of the curves, so we can say here for the C3 and then up there for the C4 plant. This represents the temperature range at which temperature is optimum for photosynthesis, so obviously optimum for the photosynthetic enzymes. And then we have the descending part of the curves, as you can see there. And the descending part of the curve is associated with temperature sensitive negative effects, some of which are reversible and others are not reversible. These high temperatures affect all biochemical reactions of photosynthesis, as well as the integrity of the membranes in chloroplasts, which is obviously a problem as well. So photosynthesis is limited here by the activity of rubisco, and the response to temperature reflects the two conflicting processes. That is an increase in carboxylation rate and a decrease in the affinity of Rubisco for carbon dioxide as the temperature rises. There is evidence that the activity of Rubisco enzyme decreases at high temperatures because of the temperature effects on another enzyme which activates Rubisco, and that other enzyme is called Rubisco activase. So these opposing effects dampen the temperature response of photosynthesis. And of course, in a C4 plant, the leaf interior is already saturated with carbon dioxide in those bundle sheet cells. So these plants have a higher photosynthesis temperature optimum than the C3 plants when grown under their natural conditions or in their natural conditions. So we've looked at how light and temperature influence photosynthesis. Now we're going to turn our attention to the effects of carbon dioxide concentration. Carbon dioxide diffuses, of course, from the atmosphere into the leaves, first through the stomata, then through the intercellular air spaces, from there into the cells, into the cytosol of the cells, and then from the cytosol into the chloroplast organelles. So if there's adequate light, then higher carbon dioxide concentrations theoretically support higher rates of photosynthesis. Low concentrations of carbon dioxide, of course, limits the rates of photosynthesis. Now, carbon dioxide, of course, is a trace gas found in the atmosphere. It accounts for only about 0.039% of air. Water vapour accounts for about 2%, oxygen accounts for about 21%, and nitrogen about 77%. And because diffusion rates of carbon dioxide into the leaf depends on a concentration gradient, then appropriate gradients are needed to ensure adequate diffusion. Keep in mind that the cuticle is nearly impermeable to carbon dioxide diffusion. So the main entry, of course, is via the stomatal pore. And of course, the same path is travelled by water vapour that is exiting the leaf in the process of transpiration. So carbon dioxide diffuses into the air spaces in the gaseous phase. The remainder of the diffusion pathway into the chloroplast is in the liquid phase, and this starts at the thin film of water on the outer surface of the mesophyll cell walls, as we saw in one of the earlier lectures. Sharing this stomatal entry pathway by carbon dioxide and water actually presents a functional dilemma to the plant. In high humidity, the diffusion gradient that drives water loss is estimated to be about 50 times larger than the gradient driving CO2 uptake. Yeah, this needs to be removed. That's a typo there. Okay, so the diffusion gradient that drives water loss is about 50 times larger than the CO2 gradient, uptake gradient. And in dry air, this difference is even greater. Therefore, a decrease in stomatal resistance through opening of the stomata facilitates higher carbon dioxide uptake, but unavoidably also is accompanied by substantial water loss. So each part of the diffusion pathway imposes what's referred to as a resistance to carbon dioxide diffusion. The gas phase of carbon dioxide diffusion into a leaf can be divided into three components, 3 points of resistance: the boundary layer, the stomata, and the intercellular spaces within the leaf. And each of these imposes a particular magnitude of resistance to CO2 diffusion. So let's evaluate the magnitude of each of these resistance points to understand the limitation to photosynthesis when we talk about CO2 diffusion. All right, the boundary layer you're already familiar with, and you know that this consists of relatively still air, unstirred air at the leaf surface, and its resistance to carbon dioxide diffusion is called boundary layer resistance. The magnitude of this boundary layer resistance decreases with leaf size and, of course, wind speed. Smaller leaves tend to have a lower boundary layer resistance to carbon dioxide and water diffusion, and also to sensible heat loss. Leaves of desert plants are usually quite small, and this facilitates heat loss. Large leaves, on the other hand, for example, large leaves found in the shade of humid tropics, can have very large boundary layer resistances, but these leaves can dissipate heat load by evaporative cooling, and this is made possible by the abundant water supply that you would find in these habitats. Next, we have resistance to carbon dioxide diffusion by the stomatal pore. And this is referred to as stomatal resistance. Stomatal resistance is generally much greater than the boundary layer resistance because when you think about it, air is very rarely still. So boundary layer, the air within the boundary layer is constantly being stirred. So the main limitation to carbon dioxide diffusion is this stomatal resistance. In other words, when you think of stomatal resistance, think about whether the stomatal pore is open or closed. Okay, next we have resistance to carbon dioxide diffusion in the leaf air spaces, called the intercellular airspace, and this is called intercellular airspace resistance. This resistance to CO2 diffusion is usually small, quite small, causes a drop of about 0.5 pascals or even less in partial pressure of carbon dioxide in comparison to the 38 pascals of partial pressure of CO2 outside the leaf. And finally, and not shown here, but we have resistance to carbon dioxide in the liquid phase, called the liquid phase resistance or mesophyll resistance. And this involves, this resistance involves diffusion from the air spaces in the leaf to the chloroplast stroma, that is the site of carboxylation. But since chloroplasts tend to localise near the cell periphery, this minimises the distance across which carbon dioxide has to diffuse. So this type of resistance is estimated to be about 1/10 of the combined boundary layer resistance and the stomatal resistance. So a minor factor. Okay, we know that leaf anatomy is specialised for capturing light and how it facilitates the internal diffusion of carbon dioxide. But this begs the question, where inside a leaf does maximum rates of photosynthesis occur? In most leaves, light is preferentially absorbed at the upper surface, whereas, of course, carbon dioxide mostly enters through the lower surface of the leaf. So given that light and carbon dioxide enter from opposite sides of the leaf, does photosynthesis occur uniformly inside the leaf, or is there a gradient? For most leaves, internal carbon dioxide diffusion is quite rapid. So limits on photosynthesis are actually imposed by factors other than carbon dioxide supply. When light enters the upper surface, the blue and red photons are obviously mostly absorbed by the palisade mesophyll cells, while green light actually penetrates, well, that green light that's not reflected penetrates deeper into the leaf. And we know that chlorophyll pigments absorb green light very poorly, yet green light is actually very effective in supplying energy for photosynthesis when the leaf is depleted of blue and red photons. So everything that we've ever learned about green light, we need to sort of relearn a little bit because there is some green light, minimum amount, but some green light that can be utilised by chlorophyll molecules to drive photosynthesis. Oh, sorry, I have a question here. What's going on? Okay, I won't repeat that comment in the recording. So when a plant is under water stress, some close their stomata. Yeah, good question. Question is, for those listening to the recording, so when a plant is under water stress, some close their stomata. Absolutely correct. Absolutely. And we're going to see some really neat evidence of that in an indirect way in the prac workshop next week. So good question. Or good statement, I should say, yes. Okay, we are nearly there. So, carbon dioxide assimilation depends to a large extent on rubisco content through a leaf, within the leaf, and distribution throughout the leaf. In spinach and fava bean, they found that rubisco content is low at the top of the leaf. It increases towards the middle of the leaf and then decreases again towards the bottom of the leaf. So similar to the distribution of chlorophyll in a leaf as shown in this graph. So we can see here chlorophyll content. Here it is here. So following this line, this black line here, and that's shown as a function of tissue depth. So this would be the upper surface of the leaf, low level of chlorophyll, lower level of chlorophyll, and then as we get deeper into the leaf, so about here, so about midway in the leaf, we have maximum chlorophyll content, and then it decreases as we get to the bottom of the leaf, but still quite high relative to the chlorophyll content right here. So that's quite interesting that they've actually determined that. So of course, carbon dioxide imposes limitations on photosynthesis. For many crops such as tomatoes, lettuce, cucumbers, for example, that is those crops grown in greenhouses with optimal water and nutrients and temperature and possibly carbon dioxide enriched environment, this increases productivity results. Photosynthetic rate can be expressed as a function of the partial pressure of carbon dioxide in the air spaces in the leaf. And this makes it possible to evaluate limitations to photosynthesis imposed by carbon dioxide supply. So let's have a look at this graph here, which plots carbon dioxide assimilation as a function of the partial pressure of intercellular carbon dioxide. And we can see some interesting features here between C3 plants and C4 plants. So in C4 plants, photosynthetic rates actually saturate very quickly at about 15 pascals. That's the unit being used. Yep, at about 15 pascals, we can see that saturation occurring. In the C3 plants, increasing carbon dioxide pressure levels, they continue to actually stimulate photosynthetic rates over a much broader range of carbon dioxide pressures. What else can we see here? In C4 plants, the carbon dioxide CO2 compensation point is 0 or nearly 0, which reflects their very low levels of photorespiration. And in C3 plants, the carbon dioxide compensation point is about 10 pascals, and this reflects carbon dioxide production because of photorespiration, which causes a loss of CO2. So just some interesting features regarding carbon dioxide pressure. Okay, and that's actually all summarised here for you. All right, so inspecting the plot on the previous slide. In C4 plants, photosynthesis rates saturate at values of 15 pascals, reflecting the effective carbon dioxide concentrating mechanism. Yes, in C3 plants, increasing values stimulate photosynthesis over a broader range. Yes, we said that. In C4 plants, the CO2 compensation point is nearly 0, reflecting low levels of photorespiration. And in C3 plants, the CO2 compensation point was about 10 pascals, reflecting CO2 production or loss due to photorespiration. Okay, so these responses that we've seen to concentrations of carbon dioxide indicate that C3 plants may actually benefit more from ongoing increases in today's atmospheric CO2 concentrations, as shown here. In contrast, because photosynthesis in C4 plants become carbon dioxide saturated at low concentrations, they don't stand to benefit much from increases in carbon dioxide in the atmosphere. All right, and that's described for you here. In fact, the ancestral photosynthetic pathway is the C3 pathway, which evolved when carbon dioxide concentrations were much higher. The C4 pathway is thought to be a derived pathway which evolved much later. Even though this is the case, photosynthesis in C3 plants is carbon dioxide limited, but still they account for about 70% of the world's primary productivity, even with their problems with photorespiration. All right, so looking into the future, an important question today is how will photosynthesis and respiration be affected in an environment where carbon dioxide levels may reach 400 plus parts per million. And this, of course, is particularly relevant as we continue to add carbon dioxide to the atmosphere. That is carbon dioxide derived from fossil fuel combustion. A promising approach to study the physiology and ecology of plants in environments with elevated carbon dioxide levels has been the use of free air carbon dioxide enrichment experiments, simply known as FACE experiments. And using FACE experiments, they can enclose entire fields or natural ecosystems in these tubes that add carbon dioxide to the air to simulate the high carbon dioxide environment that we might expect to see in the future. And these experiments are already providing new insights into how plants might respond. Some interesting observations have come out of these experiments. So one key observation is that of a 20% increase in growth of C3 plants under enriched carbon dioxide, with no increase at all in C4 plants, as expected. Another common observation in these FACE experiments is that plant growth is quickly constrained by nutrient availability, which is interesting given all the fertilisers that we use in cropping. Pictured here are FACE experiments that have been conducted in a stand of deciduous trees, and these represent mixed and unmixed stands of aspen trees in Wisconsin. And we have a comment, so trees will come back. Well, yes, well, yeah, that's interesting. So there are actually different schools of thought, even though these FACE experiments, you know, are showing that C3 plants you know, will come back and they'll thrive. C4 plants, there won't be any difference. There are sort of conflicting results, which we won't go into, but you can certainly read all about them if you do a bit of a Google search. So it's not all as clear cut as it seems. But anyway, but we're still hopeful. So, woody trees. Yeah, I'm not sure. Yeah, that statement, I'm not sure if that just relates to woody trees or other crops. But I have seen some conflicting experimental results from these experiments. All right, some more FACE experiments. These ones are being conducted in a soybean field in Illinois. And under elevated carbon dioxide levels, they found that leaf stomata were more closed, resulting in higher leaf temperatures, as shown by this infrared image down here of a soybean canopy. So here's the FACE experiment here. Temperature recorded as 27.5 degrees Celsius, and under ambient carbon dioxide, the temperature was 26.1 degrees Celsius. So this could be where some of those conflicting results come from results such as these. And that brings us to the end of the lecture. Thank you, everyone, for your wonderful attention. Who has questions? None from you? Okay, questions from anyone else? Yes, I think so. I think so. I think Luke's dropped off. Yep, his internet might have dropped out. I'm not sure if Pat's really there. Pat, are you actually there? Patrick? I might have put him to sleep. I've done that before. A student told me once that they fell asleep on slide 24 of one of my lectures. Yes, you should go and start your assignment. Absolutely. All right, I'll leave you to it. Thank you, everyone, and look forward to the prac workshop next week. It will be wonderful. All right, thank you. Good night.